Now, we will discuss about receptive fields. What is receptive field? Levine and Schaffner in 1991 defined a receptive field as an area in which stimulation leads to response of a particular sensory neuron. According to Alonso and Chen, 2008, the receptive field is a portion of sensory space that can elicit neuronal responses when stimulated. The sensory space can be defined in a single dimension, 2-dimension, or multiple dimension, and tuning properties of a visual receptive field. The neuronal response can be defined as firing rate, which are the number of action potentials generated by a neuron, or include also subthreshold activity. For example, depolarization and hyperpolarization in membrane potential that do not generate action potentials. Therefore, here, for a visual system, receptive field is the visual space or the region of space that is associated with a neuron. In the example, when a certain visual stimulus in the region is given, the firing neurons will change. Receptive fields in the retina have been found in certain layers, such as in the bipolar cells, ganglion cells, with a different response pattern, either on-center or off-center. The response patterns reflects the receptive field structure differences of illumination levels or local changes in light intensity. We will discuss this part later. Figure shows the example of receptive field in the retinal layers. Each bipolar cells, which is not shown here, receive input from many photoreceptor cells, and each ganglion cells receive input from many bipolar cells. The receptive field of each photoreceptor corresponds to the portions of the visual field with the same letter. In the example, photoreceptor A sees the snake head. The photoreceptor J sees the rattles on its tails. The receptive fields of the ganglion cells are determined by the pattern of input from the photoreceptors via bipolar cells. For example, if we recorded the receptive field in ganglion cell 1, its receptive field will include portion A and B of the visual field, which is the snake's head and the upper body. Ganglion cell 3 only gets input from photoreceptor E, so its receptor field is E. While for ganglion cells 5, it will get input from G, H, I, and J of the photoreceptor. So it sees most of the black portion of the tail plus the rattles. In summary, receptor field is defined as a patch of visual field or a group of objects or properties that shares a common characteristic which will change the firing of neuron in the region. Here we will discuss the on-center and off-center in retinal layers. The response patterns at the retinal layers reflects the receptive field structure differences of illumination levels or local changes in light intensity or light response properties. The circuit that processes the sensory input from vision or visual input is called on and off pathway. The on and off processing originates at the first retinal synapse between photoreceptors and bipolar cells. A set of photoreceptor cells, which is rods or cones, send inputs to bipolar cells. This process defines the bipolar cell's receptive field. The receiver's response at on and or off bipolar cells is upon exposure to light. In the example, in the dark, photoreceptors is depolarized and they release more or maximal glutamate and excite the off-center bipolar cells. Here, off-center bipolar cells are depolarized by glutamate. These are called off-center bipolar cells because the bipolar cells are active when the light is off in the center of their receptive field, while in the presence of light, photoreceptor is hypopolarized and they release less glutamate and excite the on-center bipolar cells. Here, on-center bipolar cells are depolarized by glutamate. These are called on-center bipolar cells because the bipolar cells are active when the light is on in the center of the receptive field. Just to add, to relay visual information to bipolar cells, there are two types of connections or pathway involved. Number one, straight-through connection or vertical pathway, in which photoreceptors connect the bipolar cells directly and convey the information to retinal ganglion cell, which is involved the receptive field center. And the second pathway is lateral pathway, in which information from photoreceptors is transmitted to bipolar cells via horizontal cells and amacrine cells, which allow other elements in the retinal layers to relay information for further processing that involve the receptive field surround. Same like bipolar cells, the response pattern at ganglion cells reflects the receptive field structure differences of illumination levels or local changes in light intensity or light response properties. Ganglion cells also has two types, which are on-center and off-center ganglion cells. Ganglion cells do not create a receptive field pattern. However, they transmit or relay the organization of a receptive field that is already present at the level of bipolar cells. In the retina, ganglion cells are the final output neurons that collect electrical messages regarding the visual signal and transmit information to the brain. Some ganglion cells receive inputs that are equivalent to a single or just a few bipolar cells. This small receptive field allows the ganglion cells to encode fine details of the visual field. Next, the axons from these ganglion cells sends the visual information all the way up to neural pathway to the visual cortex. The size and position of a light stimulus will affect the response of on-center ganglion cells and off-center ganglion cells. If we look at the stimulus A, diffuse illumination produces conflicting input to on-center ganglion cells. The center excites the ganglion cells while surround inhibit it. Because the surround has a larger area, the result is malinhibition. Off-center ganglion cells, on the other hand, off-center ganglion cells are inhibited by light in the center and excited by light in the surround. We look at another example, which is the stimulus C. On-center ganglion cells respond more vigorously to light that fills at the center and dark in surround. If you look at example E, off-center ganglion cells responds most vigorously to dark in the center and light that fills the surround. Additionally, before the electrical signals corresponding to visual informations are conveyed to the brain, such visual information undergoes extensive additional processing in the ganglion cells. In 1953, Kuffler found that each ganglion cell has a different response pattern, either on-center or off-center. The response patterns reflect the receptive field structure or retinotopic map differences of illumination levels, or local changes in light intensity. Another process that is also involved is the splitting of the visual information into specific ganglion cells. There are three types of ganglion cells, which are Megat cells, which around 80% of ganglion cells. Next is parazole cells, around 10% of the ganglion cells, and other types, which around 10% of ganglion cells. Each ganglion cell types has a different function in the detection of visual input and also forms a separate pathway to reach its target in the lateral genic nucleus. Megat cells receive inputs from cones conveyed by bipolar cells that presume color opponent response properties with a spectral selection of red-green or blue-yellow. According to De Monastrio and Goras in 1975, Megat cells have small receptor fields and allow relatively high visual acuity. In the retina, the fovea is the region that contains a high concentration of Megat ganglion cells, and therefore exclusively provides high acuity and high color sensitivity. The anatomical features of Megat cells allow them to specialize in color vision and high spatial resolution. In example, object recognition features such as pattern and texture, but with low temporal resolution. Megat cells form a projection to parvocellular layers of the LGN, the so-called P pathway. While for parazole cells, parazole cells receive inputs from a relatively large number of rods and cones. Parazole cells have relatively larger receptive field that give a low spatial resolution. The anatomical features of parazole cells allow them to specialize in low spatial resolution and provide high temporal resolution. In example, motion detection. Parazole cells form a projection to magnocellular layers of the LGN, the so-called M pathway. Another type of ganglion cells has been identified, which is small bistrophic cells, which form a projection to coniocellular layers of the LGN, the so-called K pathway. Bistrophic cells receive inputs from intermediate numbers of rods and cones and are presumed to have a moderate spatial resolution and moderate temporal resolution. They are likely to be involved in color vision.